Many bacteria have the ability to exchange DNA, and when that exchange is made, the new DNA merges with the old DNA and allows for a broader range of traits, often to the host's advantage, to benefit its survival. This is important because bacteria reproduce asexually. Without genetic variation, bacteria are more susceptible to being killed. With genetic variation, bacteria have a much higher chance of survival. Frederick Griffith used mice and various strains of bacteria to prove this theory of transformation. The bacteria used was Staphylococcus pneumoniae.
The first strain was rough and non-virulent.
When injected, the mouse lived. The second strain tested was both smooth and virulent.
When injected, the mouse died.
The third was a heat-killed and smooth strain. When injected, the mouse did live. The final strain was a mix of the rough and the heat-killed smooth.
By mixing the bacteria, it killed the mouse.
No mice were harmed in the making of this video. Next up is transduction.
Joshua Lederberg and Norton Zinder.
Then phages are created with host cell DNA, and phages are released to go inject other host cells with the donor cell DNA. Then the donor cell DNA recombines with the new DNA. Generalized is a similar process, but the coding is much more random than specialized. Zinder and Lederberg actually discovered transduction by accident. They intended to prove conjugation in Salmonella, but discovered viral unit P22 was responsible for the recombination of genes. Joshua Lederberg and Edward Tatum. In this diagram, the red represents DNA. The blue represents a donor cell. The pink is a recipient cell, and the green is an F plasmid. The donor cell attaches to the recipient cell, and the plasmid splits and shares the DNA through both the cells. The recipient cell then becomes a donor cell. A plasmid is a small DNA molecule within a cell that is physically separated from chromosomal DNA and can replicate independently. They are commonly found in bacteria. Plasmids were discovered, or the term was coined in 1952 by, you guessed it, Joshua Lederberg. This short film was brought to you by Sid and Mimi.